During this video, we are going to discuss gene expression, basically how we control what genes are being turned on and off, what RNA strands are being made. So, what you're going to learn in this video is looking at the relationship between DNA, RNA, and protein synthesis, how you control gene expression, how there are differences between eukaryotic and prokaryotic cell gene expression, and then how you actually apply this to yourself. So what actually is gene expression? It's basically the turning on of a gene and making a protein. And it only occurs when the protein is actually needed in your body, because if it's made or occurs all the time, then you're making too many proteins and wasting energy. And what ones are actually made are controlled by the cell, and it includes both transcription and translation, which, if you remember, transcription is the making of RNA from DNA, and translation is the RNA to a protein. So what actually started this whole thing was Jacob and Monod started looking at E. coli and discovered how genes control the metabolism of the sugar lactose by producing 3 different enzymes. So basically they discovered what gene expression actually is and what controls gene expression. So what they actually really found was that in bacteria there is an operon. An operon is composed of 3 parts: the promoter, the operator, and the structural gene. The operator is actually the beginning of the gene, so you can think of it as the starting line. The operator is actually a place for an enzyme to bind to that blocks transcription. So it's kind of the on-off switch. And then the structural gene is actually the code for your protein that you are making. And it goes in that order in their DNA. So what actually happens is a gene can either be turned on or turned off, repression and activation. For repression, there's no gene expression. So there is the protein bound to the operator, making it so that the RNA polymerase tries to come through and then literally gets blocked. It's a roadblock there. So therefore no transcription can occur. Now what happens when the gene gets turned on? It makes gene expression happen. And this is caused by a stimulus. So if you see in the animation, the black circles are lactose. And basically the lactose goes and binds to the inhibitory protein. That protein leaves, and therefore RNA polymerase is able to go through and cause transcription to occur for that gene. Now, as soon as there's no more lactose, then the inhibitor goes back on, and therefore shutting off the gene, which makes sense because if you don't need the enzymes anymore, if there's no more lactose, you don't need the enzymes, and therefore the gene should be turned off. So, for example, you drink milk. Your stomach breaks down the milk into lactose, and the lactose goes into your intestines, entering the bacteria in your intestines. The lactose goes and binds to the repressor protein and makes it come off of the DNA, and the lac operon genes are transcribed. So the RNA polymerase is able to go through and transcribe and then translate those enzymes that break down lactose. So the enzymes that break down lactose are produced, and therefore lactose is able to be broken down. So it's all controlling itself, which is a huge bonus of this whole process. There are some genes that are constantly turned on and actually need to be stimulated to be turned off. So the problem with this is bacteria is a prokaryotic cell, and our cells are eukaryotic. So therefore, we have much more complex DNA, and no operon has actually been found in our cells. So it's a little more complicated, but to simplify it, eukaryotic genes, there's no operon, but we do have a promoter and what's known as an enhancer, which is kind of like the operator. And we use transcription factors instead of the inhibitory or regulator protein. And we also have 2 different types of segments in our genes, which we'll look at in a little bit. So basically, When activation occurs, transcription factors bind to the enhancer and promoter, and as you see in the picture, it brings the RNA polymerase to the beginning, to the promoter, to start the coding. So the enhancer and the promoter do not have to be next to each other. In order for this to happen, they can actually bend, because if you remember, prokaryotic DNA is circular, ours is just one long strand, so it can bend and move and on top of each other. So then RNA polymerase goes and starts transcription, and the entire gene is transcribed into an mRNA strand. So what you actually get is a whole gene, and it's composed of these things known as exons and introns. Exons are actually part of your DNA that code for amino acids, and introns do not code. So that's what's known as your junk DNA. And what happens for this is it allows us to control transcription, or our cells to control transcription. Because if there's wrong coding in there, if it occurs in the introns, it doesn't affect us at all. If it occurs in the exons, while we're actually editing this, we'll be able to check. So it's a double check. So what actually happens after transcription, you have to take out the introns. So you splice them out. So if you see in the picture, your first picture is your whole gene made up of DNA, which is exons and introns, and then it was transcribed into what you call your primary RNA, and then you have to splice out the introns in order to make your mRNA, and then the mRNA gets translated into a protein. So they don't know for absolute sure why we have exons and introns, other than for the double editing, and in case there is a mutation in the introns, it won't really affect us at all. So that's the end of gene expression, the very basic level, just basically how and why various proteins are made at certain times. And it's all controlled actually by your body. You have genes that are always turned on, and then when they're not needed, they get turned off, and then genes like the lactose genes that have the lac operon that only get turned on when you drink milk. So if you think of back to insulin, we only will make insulin when we need it. So the blood sugar level actually controls and turns on the insulin gene expression. So it actually makes sense for your body to control itself that way.